Hey guys, Miss Cote here. We're continuing our unit on cell communication by looking at the nervous system. The nervous system is all about communication, and neurons accomplish this by using two types of signals. Within the neuron itself, the signals are electrical, while between neurons, the signals are chemical. In this chapter, we're going to look at the structure of the neuron and how it accomplishes the electrical part of the signaling. So let's get started. You can't talk about the nervous system without exploring the neuron. It has a specialized structure that's very easy to recognize. One of our recurring themes in this course is the relationship between structure and function, and the neuron is a perfect example of this theme. So here's a diagram of two neurons, and let's look at how it's organized. Over here on the left, we have the cell body. This is where most of the organelles of the cell are found, including the nucleus. There are multiple branches extending from the cell body. These are called dendrites. The function of the dendrites are to receive an incoming signal from other neurons. Notice the red arrows going inward toward the cell body. The longer extension coming off of the cell body is called the axon. The axon's job is to transmit information to other neurons. As you can see, the information travels in a one-way direction from the dendrites down the axon to the next neuron. The axon ends in branching structures called synaptic terminals. From this view, it looks as if the synaptic terminals are physically touching the next neuron. But if we take a closer look, there's actually a very small gap between the two neurons. This gap is called the synapse or synaptic cleft. The signal that travels within the neuron is an electrical signal, which we'll discuss later in more detail. However, when it gets to the synapse, electricity cannot jump across a space. So when the electrical signal reaches the synaptic terminal, it can't just jump across the gap to the next neuron. It has to find another way to get over there and continue the signal. This is where the chemical signals come in. The chemical signals that carry the information from one neuron to the next are called neurotransmitters. So remember that information travels in one direction. We refer to the cell that is transmitting as the presynaptic cell, pre referring to before the synapse, and the signal is going to the postsynaptic cell, post referring to after the synapse. While the presynaptic cell is a neuron, the postsynaptic cell can either be a neuron to continue the signal, a muscle cell when the end result is movement, or a gland cell when the end result is to secrete a hormone or other molecule. The processing of information happens in three stages: sensory input, integration, and motor output. Each stage of processing is taken care of by groups of neurons. Sensory input is taken care of by sensory neurons. Their job is to detect stimuli, whether external, like light, touch, or smell, or internal, such as blood pressure or muscle tension. Next, the information received needs to be sent to the brain, where it can be processed by interneurons. And then the response is sent out through the motor neurons, which will trigger muscle or gland activity. At some point, you may have learned that there are two parts to a nervous system. The brain and spinal cord would be considered the central nervous system, or CNS, and all of the nerves that branch out from them are referred to as the peripheral nervous system, or PNS. To put this in the context of information processing, the CNS consists of the neurons that are involved with integration, the interneurons, and the PNS consists of both the sensory and motor neurons. Now that you're more familiar with the structure of a neuron, let's look at how they function in signaling. In neurons, like other cells, the inside of a cell is more negatively charged than the outside of the cell. This unequal distribution of ions on either side of the plasma membrane gives it a membrane potential. When a neuron is not sending a signal, it's considered to be at rest, and this is called the resting potential. It's usually somewhere around -60 to -80 millivolts. So any changes in the membrane potential is going to act as a signal to transmit information. The two ions that are crucial in membrane potentials are sodium and potassium. Usually the concentration of potassium is higher inside the cell, while the concentration of sodium is higher outside the cell. This represents chemical potential energy. These concentration gradients are maintained by the sodium-potassium pump. If this term doesn't ring a bell, this is an example of active transport we discussed back in Chapter 7, and since it is active transport, ATP is required to maintain this gradient. ATP is used to pump sodium out of the cell and pump potassium into the cell. The chemical potential can be converted to electrical potential through the use of ion channels in the membrane. Depending on which channels are open, specific ions can cross the membrane, moving down their concentration gradient from high to low. This can change the membrane potential of the cell. The resting potential is negative because there are more potassium channels open at rest than sodium channels. So while some sodium ions can enter the cell, more potassium ions will move to the outside of the cell, ultimately making the inside more negative. Remember, the resting potential is somewhere between -60 and -80 millivolts. The cell doesn't get overly negative because the excess negative charge will attract the positive potassium ions and oppose the flow to the outside of the cell. So when a neuron responds to a stimulus, we see changes in the membrane potential. These changes happen because gated ion channels open or close in response to a stimulus and allow ions to flow into or out of the cell. This is going to alter the membrane potential. Let's go back to our sodium and potassium ions in the neuron. Remember that the potassium ion concentration is higher inside the cell than outside. So if we open potassium channels, then potassium ions will flow to the outside of the cell. This will make the inside of the cell more negative. We can refer to this as hyperpolarization. And if we were to measure the voltage, hyperpolarization would look like this on a graph, with the membrane potential becoming more negative. Also remember that the sodium ion concentration is higher outside the cell. So if we were to open sodium channels, sodium ions would flow into the cell, making it more positive. This is referred to as depolarization. And again, measuring the voltage and looking at what it would look like on a graph, it would look like this. What these two graphs are showing are small shifts in the membrane potential. These shifts are not signals. They can be referred to as graded potentials. In order to transmit an actual signal, you need a massive shift, a depolarization in the membrane potential. This is called an action potential. If you notice on the graphs, there's a dotted line above the resting potential. This is called threshold. If you depolarize the membrane enough to reach threshold, this will get you an action potential, which looks like this. In this graph, we have depolarized the membrane potential to threshold, about -55 millivolts, and we see a large spike in the membrane potential. An action potential is an all or none response, meaning it either happens or it doesn't. There's no in between. Action potentials come about because of voltage-gated ion channels that open or close in response to a change in the membrane potential. So if the membrane potential starts to become depolarized enough, this will cause the opening of these gated sodium channels, allowing sodium ions to flow in, resulting in more depolarization, which opens more channels. This is a positive feedback mechanism, with the result being a rapid influx of sodium into the cell, producing a significant change in membrane potential. An action potential can be considered as a series of changes. So we're going to break down this process by looking at what's happening in the membrane at each stage. This is a diagram that you should be very familiar with. So stage 1 is resting potential, which we've just talked about, and we can see on the graph that at rest the membrane potential is between -60 to -80 millivolts. Looking at what's going on in the membrane, we can see that most voltage-gated sodium and potassium channels are closed. We begin to see depolarization at stage 2, where the membrane potential, in this case, gradually proceeds to threshold, which is about -55 millivolts. Looking at the membrane, we see voltage-gated sodium channels open first to allow sodium ions into the cell. Remember, this makes the inside of the cell more positive, and the more channels open, the more sodium ions flow into the cell, ultimately bringing it to threshold. If threshold is reached, then the cell enters the rising phase of the action potential, which is stage 3. Remember that the action potential is all or nothing, so there's no stopping it at this point. The membrane potential rapidly increases. In the membrane, all of the sodium channels are open at this point, causing a massive influx of sodium ions into the cell. Stage 4 is the falling phase. The sodium channels close, actually very soon after opening, and most of the potassium channels now open. This causes a rapid outflow of potassium ions outside of the cell, bringing the membrane potential back down toward resting. So in the membrane, you can see that most of the sodium channels are now closed and the potassium channel is open. This will reverse the charge inside the cell. Stage 5 is called the undershoot. The potassium ions are so intense on getting out of the cell that they actually go right past resting potential and hyperpolarize a little. The gated potassium channels close and the cell gets itself back to resting potential. The remaining sodium channels close and some potassium channels are still open. These will eventually close as well. Because the sodium channels are inactivated during stages 4 and into 5, if another signal arrives, there cannot be another action potential so quickly. This is called the refractory period. Basically, a refractory period is a period immediately following stimulation, in this case the membrane, during which the nerve is unresponsive to further stimulation. It also makes sure that the signal is only flowing in a one-way direction. So what we just looked at is what happens during a single action potential. This actually happens in a series where the action potentials move along the axon. The depolarizing of one region in the axon will cause the neighboring region to start depolarizing, hopefully to threshold to continue the signal. It moves down the axon like a series of dominoes falling one after the other. For example, here we have an action potential shown in blue that's occurring on the axon. The sodium ions are flowing in and reversing the charge inside the cell. Note that the entire axon is not experiencing the action potential, just one piece of it. The depolarizing of the membrane has now spread to the next section, starting an action potential there. The spot that just finished the action potential is repolarizing, shown in green, and is in the refractory period, so it can't depolarize and fire another action potential right away. This prevents action potentials from traveling in the backwards direction. And lastly, this depolarization and repolarization process is repeated and will eventually make its way all the way down the axon. Please understand that this happens extremely fast. A neuron can produce hundreds of action potentials in just 1 second. In general, the wider the axon, the faster the speed of the signal. Tying this to evolution and natural selection, the animal that has the wider axon diameter would be faster to respond to signals, whether it's reacting to danger or reacting to catching a meal. That animal would be selected for and would be more successful in passing along its genes for wider axons to its offspring. However, in vertebrates, the diameters of axons are not necessarily wider or skinnier, so how can we increase the speed of our signals? The evolutionary adaptation to this issue is insulation around the axon. This insulation is called a myelin sheath and is made by cells called glia. There are 2 types of glia depending on the part of the nervous system they're in. Glia in the CNS are called oligodendrocytes, and glia in the PNS are called Schwann cells. These specialized cells wrap themselves around the axon so that it has many layers of membrane around them. Note that the myelin forms what looks like beads going down the axon. The spaces or gaps between the beads are called the nodes of Ranvier. In myelinated axons, the voltage-gated sodium channels are only found in the gaps between the myelin, the nodes of Ranvier. The depolarizing current during an action potential at one node moves along the inside of the axon to the next node, shown by the blue arrows, where the next voltage-gated sodium channels are. So from the outside, it looks like the action potential jumps from node to node, shown by the red arrows. This mechanism is called saltatory conduction. Saltatory refers to leaping. And this is where I'm going to end this lecture. At this point, we've looked at the structure of the neuron and how the electrical signal in the form of an action potential can travel down the axon to the synaptic terminals. But what happens next? How does the signal move on to the next neuron? In the next lecture, we'll take a closer look at the synapse and how neurotransmitters are used to continue the signal to the next neuron. So until next time, be smart and be safe.